Hi, everyone. My name is Chelsie Miller. I am an undergraduate student at San Diego State University, and I am speaking to you on behalf of the MIT Summer Research Program. I had the opportunity to work in Dr. Alex Shalick's lab in the chemistry department here, and I will be discussing with you my project on exploring colonic allergy response in irritable bowel syndrome. So what you can expect from me today is a background on my project, the aims that we had, what we found in the data, and any future directions for the project. And so I want to start by explaining what is IBS. Irritable bowel syndrome is a functional gastrointestinal disorder that is characterized by symptoms such as abdominal pain, bloating, diarrhea, and constipation. The etiology or cause of this disease is unknown. However, one of the most prominent risk factors is acute gastroenteritis. Abdominal pain and discomfort may occur after eating in some patients, and recent studies have indicated that IBS might be caused by a loss of oral tolerance or an inappropriate immune response to foods that are not typically allergenic. And so, why do we care about this? Well, IBS affects between 5 and 10% of people in the United States, and symptoms have the potential to interfere with quality of life. We have a limited understanding of both the cause of IBS and the role of certain cells in the allergenic response process, which is what we were curious to determine here. And the findings of this project will hopefully better inform the scientific community on ways to treat specific subtypes of these FGIDs, including the development of a therapy that might be able to help. So, previous research that is done on allergenic response to foods was seen in a recent mirroring study that suggested that acute gastroenteritis may be the cause of this loss of oral tolerance that I explained just a moment ago was an inappropriate allergenic response that causes IBS-like symptoms. Researchers paired a non-pathogenic antigen with pathogenic bacteria and monitored the immune response in mice with IBS. Figure 2 indicates the different treatments given to the mice and the role of mast cells in each of them. And researchers saw that mice that were treated first with this egg protein and then infected with bacteria and then reintroduced with this egg protein showed an immune response through mast cell degranulation or a response of these immune cells. And that was coupled with histamine release, which is known to be a mast cell regulator. Figure 4 further demonstrates this in human patients, where the tissue in the colon was introduced to several different allergies that you can see here. And at least one of these food allergens was present in those with IBS when compared to healthy patients. And so you might be asking yourself, what are mast cells? Mast cells are a white blood cell present in connective tissue, and they are one of the cells that responds when you have an allergy. And through this project, we have worked to analyze the data from the PREDICT trial to better understand mast cell gene expression, what might be different between IBS and other functional gastrointestinal disorders, and whether mast cells are more abundant in IBS. And so we did this by using R programming. And we first ran the data through what is called SC Transform, which is a package here. And we found about 151,000 high quality cells, and 2,000 of those were mast cells, as you can see in the purple cluster to the left of this UMAP. And then we further analyzed the data to pull out any variable genes, like highly variable genes, which just means which genes were more expressed in these mast cells. And so through our findings of this data analysis, we were able to determine a few different clusters that looked like they were mast cell gene clusters. And this was all done through IBS versus non-IBS data. And so cluster 0 and cluster 4, we worked with those to see if they were mast cells or if they weren't. And cluster 0 was really difficult to determine what was going on because, as you can see, there's not a really clear gene expression based on the strong yellow color that's absent here. But in clusters 1, 2, and 3, we saw gene expression of known mast cell characterized genes, one of them being FOS, and another being B2M, which plays a role in the inflammatory process. And cluster 4, we were able to eliminate because, without going into too much detail, it was characterizing more of a T cell type than a mast cell. So, what does this actually tell us? Not much, I'm going to be honest, but we determined here that the biology of mast cells might not be different. And that's just because mast cells are really difficult to characterize because of their abundance in several different tissues. But we do know that IBS is really common and it's growing in demand, and we are searching for a therapy. And because of all of the complicated science behind these mast cells, this project has been really challenging. But I have a newfound appreciation for the amount of time and effort it takes to become proficient in explaining not only immunology, but also understanding program languages. And I hope that I've been able to do that here today, just to explain what my project was to you all. And so I want to give a shout out to my grad student mentor, Leonard, for helping me with this project, and a great big thank you to the Shalek Lab and the MSRP program for hosting me this summer. Thank you all. Have a great day.